Hello everyone, this is Mrs. Correa. Welcome to week 1 lecture. We'll be discussing on conception and development of the embryo and fetus during this week. The audio lecture will be including only the highlights or the difficult concepts, because if the PowerPoint with audio is too lengthy, I will not be able to upload effectively into the Blackboard. So every slide in the PowerPoint will not be discussed by me with audio, but you have the detailed lecture available for you in the Blackboard as notes. This week we will be discussing about introduction to parent-child nursing, fertilization, implantation and placental development, development of embryo and fetus, fetal circulation, fetal membranes, amniotic fluid and umbilical cord, fetal development, and factors adversely affect embryonic and fetal development. Let us discuss a little bit about what is parent-child nursing. Parent-child nursing includes care of antenatal mother and her family, that is pregnant women and her family, then nursing care during intrapartum and family, that is care during labor, and nursing care of postpartum women and family, that is care of women after the baby is born and care of her family. The nursing care of newborn, that is once the baby is born, how to take care of that baby. And nursing care of sick children. And this is basically the pediatric nursing, but we like to use the word nursing care of children and nursing care of sick children instead of pediatric nursing. Now we will start with cellular division. What is gamete? A gamete is a haploid cell with 23 chromosomes. And the gametes are ovum and sperm. Ovum is a female gamete and sperm is a male gamete. And what is gametogenesis? Gametogenesis has two parts. One is meiosis and other one is mitosis. What is meiosis? Meiosis leads to development of sperm and ova containing haploid chromosomes. What is haploid chromosome? That has 23 chromosomes. Please remember that. And what is mitosis? That is formation of two identical cells of original cells with diploid chromosomes. And the diploid chromosome has 46 chromosomes. Now let us talk about what happens during fertilization. The oocyte, that is the female gamete, and sperm, which is the male gamete, mate in the fallopian tube. So remember this, fertilization happens in the fallopian tube. And during ovulation, cervical mucus increases, becomes less viscous and more favorable for sperm penetration. And during sexual intercourse, almost 200 to 600 million sperms are deposited in the vagina. And out of this 200 to 600 million sperms deposited, approximately 200 sperm reaches the fertilization site. Then few things happens. One is capacitation. That is, a glycoprotein coat and some amino proteins are removed from the surface of the sperm's acrosome. That is the little head-like portion. Then another thing happens is the acrosome reaction. The acrosome reaction, where the enzymes from the sperm's head is released, creates a pathway through zona pellucida. Then a reaction occurs to prevent fertilization by other sperms. Once one sperm enters or penetrates the zona pellucida, a reaction occurs to prevent fertilization by other sperms. So it blocks other sperms to enter the zona pellucida. What happens after fertilization? Implantation occurs. Fusion of pronuclei of both oocyte and sperm creates a single zygote with diploid number, that is 46 in number, of chromosomes. So remember this, 23 chromosomes came from oocyte and 23 chromosomes came from sperm. And now the single zygote has 46 number of chromosomes. And the zygote is propelled by ciliary action and peristalsis, and it reaches uterine cavity in 3 to 4 days. During this time, cleavage occurs. Now what happens after fertilization? Cellular multiplication happens. The cleavage happens during this time. Rapid cellular division, mitotic cell division of zygote occurs. And the zygote then becomes morula. It is developed by 3 to 4 days after fertilization. Morula is a solid ball of cells, approximately 16 cells, and enters uterus. Morula then becomes blastocyst. Once morula enters uterus, it develops an inner mass cells and forms fluid-filled cavity, and now it is called blastocyst. Please look at the figure 7.8 from your textbook. Another process happens during this time is nidation. It is the implantation of fertilized ovum into the endometrium. And nidation occurs by 10th day after fertilization. There could be implantation bleeding during this time, like some women may experience some spotting of blood, and the blastocyst buried beneath the endometrial surface during this time. Now let us talk about placenta. Placenta develops from trophoblast cells at the site of implantation, and the lacunae develops, which is a fluid-filled space from ruptured maternal capillaries and endometrial glands. And chorionic villi develops. The trophoblast form primary chorionic villi, and it is a small non-vascular processes that absorb nutritive materials for growth. What is intervillous space? Lacuna becomes intervillous space for placenta around the villi. It contains reservoirs of blood which supplies the developing embryo and fetus with oxygen and nutrients. By 4 months, placenta reaches maximum thickness and provides oxygenation, nutrition, waste removal, and hormones. And placenta also protects fetus. The placenta has a major role during pregnancy. It helps in the substance transport. The substances are transported across placenta through various mechanisms. One is diffusion, and the diffusion can be simple diffusion and facilitated diffusion. Simple diffusion transports substances, example water, electrolytes, carbon dioxide, fatty acids, etc. Through facilitated diffusion, it transports substances like glucose, oxygen, etc. Then active transport, it transports substances, example amino acids, water-soluble vitamins, etc. Then pinocytosis and endocytosis. It transports substances such as globulins, antibodies, viruses, etc. Please read in detail from your textbook. Another mechanism is bulk flow and solvent drag. Water and electrolytes are transported by this mechanism. Another thing can happen is accidental capillary breaks that facilitate transport of intact blood cells. Then comes the independent movement, which transports substances such as maternal leukocytes, microorganisms such as Treponema pallidum, which causes syphilis. And I want you to remember this: there is no mixing of fetal blood and maternal blood occurs normally unless there is a break in the system. There is no mixing. The substances are transported through the above mentioned mechanisms. Now let us talk a little bit about the placental hormones. There are four main hormones. They are human chorionic gonadotropin, human placental lactogen, progesterone, and estrogen. I want you to read the functions of each hormone from your textbook from chapter 7. Now we need to know a little bit about yolk sac. The yolk sac develops 8 to 9 days after conception. Yolk sac is essential for transfer of nutrients during second and third week of gestation. And hematopoiesis occurs in the walls of yolk sac beginning third week. By 8th week, liver takes over this function and the yolk sac atrophies and is incorporated into umbilical cord. Now let us talk about umbilical cord. The usual location is in center of the placenta and it is 57 centimeters long and 1 to 2 centimeters diameter. And the umbilical cord has two vessels, one vein and two arteries. And the fetal blood flows through the umbilical arteries and into the intervillous space of the placenta. And the blood returns through the umbilical vein and into the fetal circulation. Then you need to know what is Wharton's jelly. It protects the umbilical cord from compression. Wharton's jelly is a specialized connective tissue that surrounds the two arteries and one vein in the umbilical cord. Let us discuss now about fetal circulation. Fetal heart begins to beat and circulate blood by end of third week, that is during fourth week. During third week, capillaries develop in the chorionic villi and becomes connected to the embryonic heart through vessels in the chorion and the connective stalk. I have posted a YouTube video on fetal circulation which is available under week 1. It explains very well on fetal circulation before birth. And I want you to carefully listen to this and learn this. Fetal circulation is a very, very important topic. You have this diagram on fetal circulation in your textbook, as well as this diagram and explanation is available in the video which I posted too. So please make sure that you listen to that. I am discussing only a little bit about fetal circulation here. The umbilical vein carries blood from placenta to the fetus. And there is low PO2. It is important to maintain the fetal circulation. And placenta is the site of waste removal and oxygenation. The umbilical cord is formed from the connective stalk during fourth week. There are 3 unique shunts in fetal circulation. They are ductus venosus, foramen ovale, and ductus arteriosus. Remember and carefully listen to the video, and you will hear that there is increased pulmonary vascular resistance and pressure during the fetal life. But after the baby is born, all these three shunts, the ductus venosus, foramen ovale, and ductus arteriosus, will close, and there will be decreased pulmonary vascular resistance and pressure. So it is important to know these changes, because when we deal with certain cardiac conditions, we need to know how to relate this. And the circulation after the baby is born is explained in chapter 17, page 643. If you get time, please carefully read through it. Now let us see what is embryonic membrane. The embryonic membranes has two separate structures, and they are amnion and chorion. The amnion is the inner membrane, contains amniotic fluid, and the chorion is the outer membrane, forms fetal portion of the placenta. It contains finger-like projections, that is the chorionic villi, that may be used for genetic testing. You must have heard about chorionic villi sampling during the first trimester. And these embryonic membranes are the early protective structures. And these two fetal membranes, the amnion and the chorion, are slightly adherent and forms the amniotic sac, that is otherwise known as bag of waters. What are the purposes of amniotic fluid? It protects and cushions fetus, maintains normal body temperature, promotes symmetrical fetal growth, allows freedom of movement, and it is essential for normal fetal lung development. Remember this. What are the characteristics of amniotic fluid? Amniotic fluid first appears at about 3 weeks, and the amount is around 30 ml at 10 weeks and 800 ml at 24 weeks. Fetal urine and lung secretions are primary contributors of amniotic fluid, and it is slightly alkaline. It contains antibacterial, other protective substances such as transferrin and immunoglobulins. It also contains albumin, uric acid, creatinine, vernix gaseosa, lanigo, etc. Now let us move on to the human growth and development. We'll be discussing about pre-embryonic, embryonic, and fetal periods. I'll be discussing very briefly on this video, and a detailed description is given in the notes, as well as please read your textbook. The first stage of development is pre-embryonic period. This is the first 2 weeks after conception. Rapid cellular multiplication and differentiation occurs during this period, and establishment of embryonic membranes and primary germ layers occurs during this period. The next stage is embryonic period. It is the period from 3rd week after fertilization through end of 8th week. And it is also called as organogenetic period, that is the formation and differentiation of all organs occurs during this time. The more discussion on week-wise development and changes happens during embryonic period is given in my lecture note. It is very easy to understand. Also, it is available in your textbook. The next stage is fetal period. From 9 weeks until birth or termination of pregnancy is called fetal period. Rapid body growth and differentiation of tissues, organs, and systems occurs during the stage, and it is a less vulnerable stage to teratogens. Remember, the embryonic stage is more vulnerable to teratogens and fetal period is less vulnerable to teratogens compared to embryonic stage. And please read more information from my lecture notes, and from the book a detailed description is given. It is easy to understand. Now let us talk a little bit about the threats to embryonic and fetal development. There can be so many chromosomal abnormalities and teratogens occurs, and for example, alcohol can lead to fetal alcohol syndrome, rubella can cause cataract, tetracyclines can stain teeth, medications and other substances such as vitamins, alcohol, tobacco, caffeine, drugs, radiation and lead exposure can lead into so many teratogenic effects. Torch infections, it is very big. Read from page 208 from your textbook. And I am not including so much discussion on this in my lecture note and audio. This is our discussion topic. So please make sure that you read all of the posts, and so that you can be an expert in this area. And also, if questions come from this topic, you will know that it is included in the discussion. And so remember to read all the posts. And thank you for listening.